So next slide, please. All right, Leah, are you on the line?
Yeah, I'm here. Can you hear me?
Great.
All right.
Go ahead.
Hi, everyone. Thank you so much for being here today. I apologize if you can hear the sound of the landscapers in the background. Today, I want to talk to you about a pilot study project on hard clam restoration in Pine Island Sound. Next. Next slide. Thank you. Hard clams, in my opinion, are very underrated bivalves. For those of you who don't know what a hard clam is, although I'm sure most of you are familiar with them, they're basically the main ingredient in clam chowder. But while they're still alive, they can be found in seagrass ecosystems buried in the sand or on the surface as large, white, round shells with prominent ridges. Hard clams have similar ecosystem roles as oysters in that they are both filter feeders. Hard clams can improve water quality by reducing seawater turbidity by filtering phytoplankton from the water and increasing the transportation of nutrients from the water column to the benthos. This benefits seagrass by increasing the light availability and reducing competition for nutrients with phytoplankton. As hard clam shells accumulate on the benthos, they can create structure, which can attract other organisms and increases biodiversity where large, healthy populations of clams exist. Hard clams are a prey for a number of species, including whelks, crabs, skates, and rays, and thus they transfer phytoplankton primary production to higher trophic levels. Next slide, please. In the past, hard clams were very abundant on the coast of Southwest Florida, and we know this from evidence left from the Calusa Indians in the form of shell mounds. So the image at the top left shows Mound Key in Estero Bay as a LiDAR image. This is an anthropogenically formed island that was once the capital of the Calusa Kingdom. So they collected hard clams and other mollusks, consumed them, and then disposed of them regularly or for larger ceremonial events. So this archaeological dig of Mound Key, they found that it was dated back to 300 to 500 AD, and they did this using hard clams. So we know that hard clam populations have been established here for a very long time. In fact, one of the largest hard clam fisheries in the world in the early 20th century was in the 10,000 Islands, where a 40-mile long, 5-mile wide swath of hard clams existed within thriving seagrass meadows. But by the mid-20th century, the hard clam fishery had collapsed due to overharvesting and changes in water quality. Next slide, please. There are two species of hard clams in Southwest Florida: the northern hard clam, Mercenaria mercenaria, and the southern hard clam, Mercenaria campechiensis. The northern hard clam has a range that is along the Atlantic coast from the Gulf of St. Lawrence in Nova Scotia to the Gulf of Mexico. The southern hard clam has a range from New Jersey to the Gulf and throughout the Gulf of Mexico. The southern hard clam is the native species to this region, but northern hard clams were introduced with aquaculture in 1998. Where aquaculture exists in the Gulf of Mexico, there are northern and southern hard clams which easily hybridize to form fertile offspring. While the northern hard clam is more valuable commercially, basically just because it has a longer shelf life, the southern hard clam is a native species to the Gulf of Mexico. It also has faster growth rates, which makes them ideal for ecosystem restoration and management. So if I orient you to the graph on the right here, this is from a study by Arnold et al. In 2009. They collected genetic samples of oysters from the east and west coast of Florida, and this graph is just focusing on our region. The black, white, and gray bars represent different genotypes of the Mercenaria. So you have the northern hard clam in black, the hybrids are white, and southern hard clams are gray. And if you just look at Pine Island Sound, you can see an increase in northern hard clams after the introduction of aquaculture in the area. And if the two species continue to hybridize here, the southern hard clam will end up becoming a rare genotype and the hybrid populations will increase. Next slide, please. Next slide, thanks. So in order to understand the potential for hard clam restoration in this region and how it will improve quality and restore valuable ecosystem services in Pine Island Sound, which is an area that is prohibited for hard clam harvesting, we propose a pilot study to evaluate the effectiveness of restoration in this region. So the green star on the map shows the area where we want to do the restoration. We're going to put about 12,000 hard clams out in a 900 meter squared area. So that's about 13 clams per meter squared. And it's really important to have a high density of hard clams since they are broadcast spawners and the gametes do need to meet in the water column in order for reproduction to be successful. We're going to monitor survival, reproduction, and water quality for 1 year after we plant the clams to see if it's successful. Next slide, please. So we're going to monitor several aspects of the hard clams to see how they respond to being planted in Pine Island Sound. We're going to go out every month to monitor survival and take growth measurements. We also collect several clams during each trip to study their reproductive cycle and determine their condition index. We will also determine the sex of the clams to estimate the sex ratio of the population. Hard clams are sequential hermaphrodites, so they start out as males and they switch to females as they get larger. Since we are starting with younger clams, we might expect the ratio to start off with more males and increase to a 50-50 male to female ratio as time goes on. The gonad ripeness is a rank based on the visual attributes of the gonad tissue smears, which you can see 2 pictures of these on the slide. On the left is the male, and if you look really closely, you might be able to see some small rod-shaped sperm on the slide. So if there's a lot of sperm or a lot of eggs, for example, the female one is the circular objects are the eggs, then we can determine when they might spawn or if they've already spawned. So gonad ripeness and condition index can be used to assess reproductive potential and fecundity of planted populations. Furthermore, monitoring gonadal development can increase our understanding of the gonadal cycle in hard clam populations in this region. Next slide, please. In this pilot study, we will determine the impact that hard clams have on phytoplankton community structure and water quality. Hard clams can reduce phytoplankton biomass and alter phytoplankton community structure, potentially reducing the relative abundance of harmful algal species. We'll use a flow cam, which is essentially a microscope that takes individual photos of each plankton through a flow cell to characterize the phytoplankton community before and after restoration. Water samples will be taken from each site to measure nutrients in the water column, and we'll also take in situ measurements of water quality parameters. Next slide, please. So we've already gone out and done 1 preliminary site survey. We picked 2 sites, and one was sort of a good site for restoration. It had muddy sand, it had a lot of tube worms in the area, and it only had about 60 shoots per meter squared of thalassia. It was adjacent to an oyster reef, and there were light epiphytes growing on the seagrass. The control site was a little different. It had shelly sand, it had a lot more seagrass, including thalassia and haliduli, with no epiphytes. The sites were about 100 meters away from each other. Both sites had a lot of similarities. They had low predator densities. We only found 1 whelk at each site. There was a lot of macroalgae present, including Hinxia, Gracilaria, Dictyota, and Ulva, and the sites were about 2 meters at low tide. However, they could potentially be exposed during king tides, and the nutrients at the sites were approximately equal. Next slide, please. So our next steps are to obtain our special activity license from the FWC, and then we can start putting clams out. We are also going to do some additional site surveys prior to the clam release with our farmer, Carter Davis, who is growing the clams right now. And he's just going to validate that the sites are good sites for hard clams to live in. And we need to receive our clams from our farmer, and our donors, the Sarasota Baywatch, donated the clams to us. Next slide, please. So thank you to everyone at the Marine Lab, the SCCF Marine Lab staff for helping with logistics and fieldwork, and to Sarasota Baywatch for their generous donation of 12,000 clams. And especially thanks to Larry Stoltz and Ernesto Lasso de la Vega for helping me organize this project and our funding from the Sanibel Captiva Shell Club. So thank you so much for listening. I will take any questions.
Great, thank you very much. That was wonderful. And we are now back to using Menti. So for those of you online, please go to menti.com and use the code displayed at the top of the screen to enter your questions. And now we'll turn it back to the presenter to answer as many as she can.
Thank you. So the first question is, do you recommend restoration with adult clams or larvae like some groups are doing now? I think it has to do with a trade-off. With adult clams, when you plant them, you're a lot less likely to get predation. So the bigger they are, the safer they are from predation. But with larvae, you're going to be able to get a lot more out there, and they're also going to be probably easier to obtain and less expensive. So have you looked at the uptake of nutrients by clams? Any experiments that quantify their nutrient removal efficiency? So my understanding of how clams remove nutrients from the water column is that they are actually just taking them from the water column, like in the form of, dissolved organic matter or phytoplankton, and they take them and they move them. They either store them in their body or they eject them as pseudofeces. So if that's something that they don't want to eat, they eject it as pseudofeces, and that goes into the sediment, and that's where a seagrass can benefit. So the seagrass is getting the nutrients directly where they need it in the sediment, as opposed to in the water column where it's available for phytoplankton or even harmful algal blooms. So what habitats did hard clams historically live in? Hard clams historically live in thriving seagrass meadows. That's their preferred ecosystem. Are certain substrates more suitable for clam restoration than others? That's a great question, and that's something that we want to look into more, but it's not something we really know. My opinion on this is that softer muddy areas without seagrass are a really good area for clam restoration, because you can monitor it really easily without disturbing a seagrass environment. But they do tend to grow better in seagrass. So it's kind of a trade-off, and it's a decision that you have to make when you're thinking about restoration. In the past, were they harvested for anything other than food? Yes, the Calusa, or the Native Americans, they used the hard clam shells as tools. Do clams prefer certain types of seagrass beds? I don't know, but I think they are, at least where we are, they're typically found in thalassia beds. Will you be monitoring relationships to seagrass abundance? That's not something that we'll be doing for this pilot study, but it's something that we want to do in the future. Do you think that the hybrid clams might be more resilient to warming water temperatures? I think that the range of the northern hard clam is definitely a more temperate range. So they do better in colder climates. Their hot spot for their range is actually like the New York, kind of New Jersey area. So I would hypothesize that they would not be more resilient to warming water temperatures. Hybrids are also more susceptible to a certain type of disease that hard clams get. So hybrids are not great for the environment or for populations. What environmental factors affect shell strength or thickness? So hard clams grow at like an optimum salinity level around like 30 parts per thousand. So, and decreasing salinities are not good for them. I also think that ocean acidification might be something that affects shell strength or thickness as time goes on, particularly in the larvae part of their life cycle. It would be interesting to see a clam seagrass habitat restoration similar to Jolene's presentation earlier. I agree. How are you measuring survivability and mortality? So we're going to put out 3 transects at each site and basically do maybe 10 quadrats along each transect and basically just get an average for what the density is, and we'll kind of use that to track survivability over time.
Thank you, Leah. That's